Neurology is the study of nerves. Nervous system is originated from ectoderm. So germinal layers, embryonic germinal layers consists of three layers, namely ectoderm, endoderm, and in the middle mesoderm. Whereas nervous system is originated from ectoderm. Nerves and their regeneration will stop after the people become adults, but in the case of hippocampus in the brain of human being and olfactory bulb, neurogenesis will continue even after adulthood. Neuron is the structural and functional unit of nervous system. It is also called as nerve cell. It consists of basically three parts, namely cyton, axon, and axon terminals. Axon is also called as cell body. It consists of nucleus, dendrite, and nasal granules in the cytoplasm. Axon is a elongated structure from the cyton. It is covered by myelin sheath and with gaps called nodes of Ranvier. Finally, axon has axon terminals. Cytons are mostly found in gray matter and axons in white matter. Brain consists of gray matter, peripherally gray matter, and in majority white matter. Cytons are concentrated in gray matter and axons in white matter. So reflex arc, it is the arc from receptor organs to or effector organs to effector organs. For example, here pin prickling is the stimulus which is received by receptor cells in the skin. From receptor we have sensory neurons. They carry sensory neurons or effector neurons. They carry messages up to the spinal cord. From the spinal cord, motor neurons carry information to effector muscles to take action. So, sensory receptor cells, sensory neurons, motor neurons, and effector organs together form an reflex arc. Neurons are of different types. Neurons with simply cytons are called axonomic neurons. An axonomic neuron consists of no axons, only cyton and dendrites are present. Bipolar neuron, here one neuron consists of two axons on both the poles, so we call it as bipolar neuron. In unipolar neuron, from single pole of the cyton, two axons originate. We call this as unipolar neuron. In multipolar neuron, from cyton and its different poles, many axons will originate. We call it as multipolar neurons. Synapse is a micro space between two neurons where acetylcholine is secreted as a neurotransmitter. So this is a synapse between two neurons. Synapse may be of different kinds. For example, here Oxodendritic synapse is there, axon of one neuron and dendrite of another neuron, that is called oxodendric synapse. And here axosynaptic synapse where axon of one neuron is having attachment with this cyton directly. We call it as axosomatic neuron. Along with the nerve cells, nervous system consists of glial cells. They are nourishing cells. They are nutritive cells. They are very important for in association with neurons to carry out the functions. There are different kinds: Schwann cells, oligodendrocyte, microglial cells, ependymal cells, astrocyte. Once again, glial cells are of different kinds: Schwann cells, oligodendrocyte, microglial cell, ependymal cell, and astrocytes. These neuroglia or glial cells have different locations and functions. For example, satellite cells, they are at a ganglia of a peripheral nervous system. They regulate microenvironment of neurons. Whereas astrocytes, they are mostly in central nervous system. They regulate microenvironment of neurons, scar tissue in central nervous system. Schwann cells, they are in peripheral nervous system. They are useful for myelination of axons, structural support for non-myelinated axons. Oligodendrocytes, they are also in central nervous system, useful for myelination of axons and form structural framework. Microglia are in central nervous system. They act as phagocytes of the central nervous system. Ependymal cells in central nervous system assist in producing and controlling composition of cerebrospinal fluid. See, ganglia of peripheral nervous system consists of satellite cells. Peripheral nervous system consists of Schwann cells. Central nervous system consists of astrocytes, oligodendrocytes, microglia, and ependymal cells.